Hello, hello.
Sir,
Yes, can you hear my voice? My voice audible
yes,
ఉన్నది?
sir.
Okay, right.
Yes, sir.
Okay. Yes, good morning to all of you, and hope others will also join soon. Yes, uh, let us start today's class. I hope all of you are safe and good at your home. Uh, okay, can you all see the slide? Slide కనబడతా ఉంది అందరికి? Anybody respond?
Yes, sir.
Yes.
కనిపిస్తుంది సార్
Okay,
స్ట్రక్చర్
right.
ఆఫ్ టిఎంవి
Yes, yes. Okay, right. Yeah. Okay. Yes, okay, dear students, in our last class, we were discussing about the virus chapter, in which up till now we have discussed regarding the virus, brief history, then we discussed about the morphology of virus based on their host, then based on their structure, different types of viruses we discussed. Then we discussed about the few virus particles like the viroids, prions, and so on. And then we discussed in our last class with regard to detailed structure of tobacco mosaic virus. Then we discussed about structure of bacteriophage morphology. Okay. And yes, today we will be discussing about the replication part, like how the bacteriophage replicates itself. How do they multiply? Yes. So normally, if you see, so reproduction it occurs in three types. Can anybody say what are the normal mode of reproduction? Organism takes place by three modes. What are they? What are those three modes? Can anybody name them?
what లైఫ్ సైకిల్ ఆఫ్ బ్యాక్టీరియా ఫేజ్
Yes, that is there. I'm asking నేను ఏం అడుగుతున్నాను నార్మల్గా రీప్రొడక్షన్ అనేది త్రీ టైప్స్లో జరుగుతుంది ఎస్ సో ఆ త్రీ టైప్స్ ఆఫ్ రిప్రొడక్షన్ ఏంటి ఎస్
Sir,
ఎనీబడి ఎస్
vegetative reproduction,
ఎస్ వెరీ గుడ్
asexual,
ఎస్ వెరీ గుడ్
సెక్షువల్
sexual.
and sexual reproduction. Yeah, very good.
Yes.
So normally three modes of reproduction we can see. One is vegetative reproduction, which the offspring is developed from the parental body part. Uh, from itself it develops. Okay, that is vegetative type. So in plants we have seen so many like from buds and so buds, bacteria, primary fission. So normally that is a vegetative reproduction and sorry, not that. So in plants, normally any part of the plant, if it regenerates, it detaches from the parental plant and it regenerates into the plant. We call it as vegetative reproduction. Then the asexual reproduction. See, normally the asexual reproduction is taking place by spores. Yes. So from spores, the uh, next generation is there. Okay, by fusion of spores. Okay. Then the sexual reproduction. See, that is the fusion of gametes. Yes. Because of that, zygote is developed and the next generation is. So that those three modes are there. But here in virus, if you see, so as it is a virus, it is not independent structure. Yes, has to be as it is dependent on a host. So there are several hosts of phytophages, zoophages, bacteriophages, cyanophages. Yes, so many different based on the host we saw different viruses. So here in bacteriophage, you see this virus, bacteriophage virus is there. Its host is E. coli. Yes, E. coli, Bacteria coli, bacteria. In its host, what does this virus do is it completely destroys the genetic material of the host during the time of replication and it takes over the total mechanism of the host and it multiplies itself inside the host and that is how its population is increased. So normally here a virus is the master. Yes, before that if you see the life cycle of virus that is bacteriophage, there are two types of life cycles you can see in bacteriophages. Yes, the first one is the lytic cycle, and the second one is isogenesis. Yes, this. So here, if you see the lytic cycle, here what happens? Virus is there, is the master, yes, because it controls the mechanism of the host, and the bacteria is the slave, master-slave relationship. Yes. So whatever master says, slave has to do. So like that sort of relation. So here the phage replicative cycle, which results in the death of the host cell, is called lytic cycle. So what happens is once the virus it takes over the bacterial total mechanism after a period of time when the virus it is completely developed. At that time, what happens here is the death of the host cell occurs. The host cell is completely dead. Yes. So what are the mechanism steps that are involved in lytic cycle is the step one will be the adsorption, call it as adsorption or attachment phase. Then the second phase we call it as penetration. Then the third phase we call it as biosynthesis phase. The fourth phase here is the assembly and the fifth phase is the lysis. So here these are the five steps. So here in the first step you see here, this is the bacterial cell. Yes, this is the E. coli bacterial cell, which has this is the bacterial DNA. Okay, the genetic material that is clear. Yes, the DNA is naked bacteria, since it is a prokaryote. So here and this is the bacteriophage virus. So this bacteriophage virus, its tail plate, if you remember, hexagonal tail plate, there are tail pins. So these, with the help of these tail pins, they have some receptors where they land on a particular site of the bacterial cell. They recognize the site and they land over there. And there, what they do is they release, they fix their pins. Okay, they fix their pins in the bacterial cell wall. attaches completely. And what do? They release an enzyme called lysozyme. Yes. So in the second step, if you see, once the attachment is done, the bacteriophage will come and land on a particular place, particular site that is on the cell wall of the that is E. coli. Second, in the second step, what happens? Penetration. It releases the lysozyme enzyme. Yes. So after releasing the lysozyme enzyme, what happens here is because of the action of that lysozyme enzyme, a hole is created in the bacterial cell wall. See, a rupture is created. And what happens here? Whatever the DNA bacteria, so this is the bacteriophage DNA, yes, the virus DNA. So this virus DNA will penetrate inside the phage DNA, sorry, bacteriophage, sorry, bacteria cell from the bacteriophage uh, particle, head region. So where is the DNA present in the bacteriophage? In the head region. So what will happen here? The tail, remember the tail is made up of capsid, contractile capsid. So as it compresses itself, creates a pressure, and that is how the DNA is injected into the bacterial cell. Yes. So yes, just 1 second. Okay. Yes. So now 1 second. So here, what happens here is because of that contractile sheet, it compresses that capsid and it creates a pressure, and that is how the DNA is injected in the bacterial cell. So this is called penetration, second step. Yes. Now in the third step, what happens here is this is the actually biosynthesis phase where you can call it as eclipse phase also. Yes, we call it as eclipse phase, Grahana Dashan. So here what happens? This once the bacterial DNA enters the bacterial cell, this DNA, this bacterial DNA will completely be destroyed or it is completely disintegrated. And what happens here? This is the viral DNA, bacteriophage DNA, which will completely control the mechanism of the bacterial cell. And it by the process of replication, what happens is viral DNA will multiply and it will create multiple copies of this phage DNA. Simultaneously, the other part of the bacteriophage, like the protein part, head region, tail region, all these are also multiplied. Multiple copies are created here. Yes, but if you see here in this stage, nothing is, you cannot see a complete mature bacteriophage particle. Yes, all the nucleic acid part, that is the DNA copies you will find multiple copies, as well as you will also find the multiple copies of the protein parts like the head region of the bacteriophage, the tail region of the bacteriophage, and so on. So nothing is clear here. That's the reason we call this biosynthesis phase as eclipse phase also. Now, if you see here, the assembly phase, yes, that is the fourth stage. This is the self-assembly phase. Now, what happens here is here all the parts, the head region, the tail region, the nucleic acid, DNA, or everything will be incorporated or it will be assembled and complete bacteriophage. You can see individual bacteriophages are uh, created, assembled. So this is just like a automobile factory where suppose there is a car factory where here the assembly phase, all the body parts of the car, like the body, the doors, the, the windows, the seats, the tires, and everything are manufactured at different locations. And here in the fourth step, assembly unit, what happens here is all the body parts are assembled, like the doors are assembled to the body part, the seats are fitted, the windows are fitted, the steering, engine, tires, everything is fitted and complete. That a car is completely ready for launch. Yes, so that's the same thing happening there. Bacterial phase also in the fourth step. What is happening here? The cell assembly phase where the all the virus particles, bacteriophage body parts are there, like protein, head, tail, DNA, and everything are assembled. Yes, so this is the assembly stage. So, what is the capacity of this assembly phase? Is somewhere around every bacteria cell, somewhere around 150 to 200 bacteriophages are multiplied. Yes, that is how that is this is the number data. This is the maximum capacity of the bacteria cell. So, this is the bursting stage problem. So in the fifth step, what happens here is during the lysis phase, same lysozyme enzyme, the effect of lysozyme enzyme, the cell wall is ruptured here, and all the bacteriophages are released at the time. Okay. This is the lysis phase. All the bacteriophage are released, and this is how from single bacteriophage we are getting multiple copies of these bacteriophages. So this is the lytic cycle, lytic phase, you can say, case of replication of this bacteria. Another cycle that is there, it is a lysogenic cycle. So this is guest-host relationship. Yes, first one we saw it was master-slave relationship, but this one is guest-host relationship. So here how that is the lysogenic cycle allow the replication of phage gene without destroying the host. In the earlier case, we saw that as soon as the bacteriophage attacks, attaches the bacteria cell, the death of the host cell will be occurring. But here, uh, this in this lysogenic cycle, the bacterial cell will not die immediately. Yes, it will allow the replication of the large genome, only genome. Yes, the DNA part. So that will be there without destroying the host. Host will be there. So what are the steps? The first step is the common step, that is the attach. So here on the bacteriophage, the bacteria, the bacteriophage come attaches. That is the first. Now what happens here? The second step is penetration. So it will release the lysozyme enzyme and it will create a rupture or hole in the bacterial cell wall. And what happens here is large DNA. So the large DNA is released here. Now here along here this is bacterial DNA. Yes, this is bacterial DNA and this is the virus DNA. So penetration occurs. So phage DNA is injected in the bacterial cell. Now what happens here is this DNA will be incorporated in bacterial DNA, bacterial gene. So here this is blue color is bacterial DNA and red color virus or bacteriophage DNA. So with the help of it will recognize the site and with the help of ligase enzyme ligation occurs and this phage DNA will be attached or incorporated into the bacterial DNA. Now this genome, this DNA has proved this stage where viral DNA attaches to the bacterial DNA. So this is the third step, that is the integration step. Yes, what is happening here? The phage DNA is integrated into the bacterial DNA. Now this bacterial DNA, what happens here is it will perform the regular cell division process. As bacteria reproduces, 1 second, somebody is waiting here. Yes, one thing I want to say here is when we are having the class at 10 o'clock, so all of you must be ready by 10. But join 5 minutes early itself. Okay? Uh, if you join afterwards, you will be missing the part. So try to be in time. Join the class in time, 5 minutes before the scheduled timing of the class, so that you don't miss it. So if you join in between, this definitely it is an interruption, and you also will not be understanding what is going on. So from next class onwards, I suggest you to join time. Yes, try to follow it. Yes. So here what happens after the bacterial large DNA incorporated? So regular normal cell division process occurs. Bacteria, what are the modes of reproduction? Sexual reproduction occurs by two methods. One is the binary fission method, another is the endosperm. So binary fission is the mode where it will perform where D is performed in the favorable conditions, where all the conditions are normal. At that time, binary fission occurs where from one mother cell two daughter cells are produced. But case of endospore, if you see in bacteria, that it is the stage or it is the type of asexual reproduction which occurs during the unfavorable conditions, develops this very thick spore wall coat outside the spore and it remains in the darkness. That is the second stage. So let us continue here that when the after the incorporation, bacterial cell what it does is it does not die immediately. Even the bacterial DNA will remain as it is, and it will undergo binary fission, the bacterial sexual death. So what happens from the one mother cell? The uh, two copies of daughter cells are produced. So these are the daughter cells. So here in both this case, you see daughter cell, you find bacterial DNA as well as the phage. Yes. So like this, binary fission continues for some time. That is how the phage genome will be multiplied. Okay. So after over a period of time, what happens here is further this recombinant DNA or hybrid cell, bacterial cell, well, that continues the lytic cycle. So this lytic cycle is continuous. So on, it will again undergo step 3, that is the biosynthesis process, where all the parts will be multiple copies of the gene, that is bacterial gene, bacteriophage gene. The protein parts of the head, the tail will be generated. And at this stage, the bacterial, what is that? The bacterial genome will be destroyed. Yes. Now the phage DNA will take over. The biosynthesis stage occurs where multiple copies of all the bacteriophage parts are developed, and after that, the fourth stage, assembly stage, will occur, where all the bacteriophage parts will be assembled and bacteriophage is ready for release. And in the fifth stage, here in the fifth stage, lysis phase, where the maximum capacity of the bacteriophage are developed to the release first. So the bacteriophage are released. So this is how the lysogenic phase occurs. So here it is just like a guest host relation. Means that the host bacteria and the guest is the bacteriophage, where it once it enters the bacterial DNA, it will remain for some time and it will keep on multiplying along the bacterial cell and the bacteriophage are developed. Yes. So this is the both the cases. You see, first case the bacterial cell dies, but here bacterial cell will not die immediately. After some period of time, then only it will be uh, continued. Second, yes. Now in this picture also you can see the attachment phase. This is the lytic here happens here. This is the bacteriophage. This is host cell. This is the bacterial DNA. First phase, the attachment phase attaches to the surface of the host. What is happening here? Penetration is occurring where the phage entered host DNA is there that will be destroyed. Okay? Penetration phase, phage viral DNA is the third phase is biosynthesis phase where the protein parts, the head, tail will be developed, the phage, all multiple copies. Nothing is clear, but this is the eclipse phase. You can see. The fourth phase is assembly phase or maturation phase where every single bacteriophage is developed. Yes, complete. So it is somewhere around 150 to 200 capacity of the cell. And the lysis of the cell occurs, the cell lies releasing the nucleus. Okay? So all the bacteriophages are released. The cell is complete. Here you can see on the left side is lytic cycle, right side is the lysogenic cycle. So lytic cycle we saw, yes, and here the lysogenic, if you see, the first step is the attachment stage where bacteriophage will attach itself to the bacterial cell. Then the second stage is strong penetration with first this is the third stage. The bacteriophage DNA is incorporated into the bacterial DNA. This is called prophage stage. And this cell multiplies, creates by binary fission, cell division occurs, and even the phage gene is multiplied. And after some time, this phage gene will be separated from this bacterial genome will be destroyed, and this will continue this phase. Yes, from here continues the lighting cycle. The eclipse phase, maturation phase, the release. That is how it goes. So that is about the replication part. Now coming down to the some viral diseases, if you see that are caused by viruses, plants, human beings. So in plants, find this is the tobacco mosaic virus, tobacco mosaic disease, then have cauliflower mosaic disease, sugarcane mosaic, leaf curl of papaya, chili mosaic, yellow mosaic, leaf rolling, leaf bleeding. These are the few diseases that are caused by virus, the plants, as well as also we find the foot and mouth disease. Uh, we find the rabies disease that are caused by viruses. Humans have common viral diseases like the common cold, flu is there, hepatitis B, SARS, AIDS, mumps, polio, dengue, chickenpox, smallpox. Yes, all these are viral diseases that are caused. Now, if you see in plants, these are the viral diseases where this is the tobacco mosaic virus, tobacco leaf, that is how it appears. Cauliflower mosaic also, you can see mosaic disease on parts of cauliflower is affected. Okay, the mosaic pattern is observed or dot 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 structure. Barley, yellowing dwarf. So the leaves are barley plant leaves are it has a stunted growth, dwarf and the leaves are yellowing. This yellowing is done by virus. Then the bud blight, where the bud is infected. Then the sugarcane mosaic, sugarcane, so sugarcane is has a mosaic pattern. Then the lettuce mosaic, lettuce also. This maize mosaic disease. This is the peanut struck virus, where the leaves of the ground are affected. Leaf curl virus, where the leaves will be curling of the leaves will be occurring. So this is how it looks. Yes, like all dot dots. The leaves, you can see mosaic pattern. This is the tobacco curl, where you can see the leaves are curling. Yes, curling of the leaves are occurring. This is papaya leaf. Papaya plant also you can see the leaves planting apart are curling. This is chili leaf curl. Chili plant also you can see curling of the curling. Tomato leaf curl. Cotton leaves are so leaf curling disease, which is caused by different viruses. Then here we have the vein clearing disease, where you can see here even vein clearing is like patterns. Okay, this is occurring completely free. Yeah, here you can see the leaves are curled. These are the symptoms. So leaves now it are completely curled. Yes, this is vein clearing. This is irregular chlorotic spots. Fluorosis where the spots are it's not like in mosaic type dot dot sort of see, but the it will be spots will be bit bigger. Okay, fluorotic. So here in vein clearing, what is happening? All the veins that are there that are completely discolored or decolored, completely yellow. The whole veins only will be get damaged. So veins are very much necessary for transportation. Whatever water, mineral ions, food are there. because it consists of all the vascular tissues, the xylem, phloem. So that vascular tissues will be affected and thereby proper supply will be not there to the beans. Water and slowly the beans will be discolored, cold and hard damaged. This is the potato leaf roll virus. Potato plant rolling is occurring where upward and upward rolling effect is occurring. This turn is whirling sort of plant growth is dwarf. Leaflet, leaflets are thick, dry, lower leaf is brown, and smaller size tubers. Potato tubers that are developed are damaged. They will be of very small size. So these are the symptoms of potato leaf roll virus. What is the transmission? That is planting of this plant, the potato, infected potato stem tubes. These are used for propagation, then the infected seed tuber that itself will transmit the disease. There are some insects like the aphid, the apis was the persistent because of insects also, aphids, they transmit the disease from one plant to another plant. That is green field. I just perceive. And if you see the foot and we saw that this foot and mouth disease is there. Here, the foot and mouth disease is highly contagious. Yes, viral disease of domestic cloven and many wild animals characterized by erosion. So, what are the regions that are infected? The mouth region, yes, mouth region of the cattle, even the hoof region of the legs, the pigs also you can see hoof region is infected. Yes, that is the mouth. These pictures where the mouth is damaged. This small face sort of thing. Region, this is the hoof region, how it is damaged. You can see. So all this is caused by foot and particular viral disease that is spreading. The humans also, there are so many viral diseases that are caused in this. If you see the common, the positive agent is the rhinovirus. The site of infection, you see, is respiratory tract, and the mode of transmission is droplet infection. Whenever somebody sneezes, so in air there's that the droplets will be spread, and if the person inhales droplets inside, that is always transmitted. So if you sit beside a person who is having cold, you will also catch cold easily. Then what are the symptoms? Nasal congestion, discharge, nose running. There you have sore throat, throat pain is there, cough is there, headache is there. So these are symptoms of common. Second one is the mumps. So this is by mumps virus, that is RNA virus, carrying virus. That is the positive agent. So what is the site of infection? The salivary gland. Mode of transmission, saliva droplet. Yes, saliva is spread. That is droplet. What are the symptoms? Enlargement of parotid glands. Parotid glands, swelling sort of things. The third one is so it is the caused by rubella virus, RNA virus category, and So virus, site of infection, skin, the skin is infected, skin disease is spread, cells and the respiratory tract infection. Mode of transmission, droplet infection. Symptoms are sore throat, running, yes, cough and fever. You'll find reddish patches in red patches will be seen where you're having some itching sensation will be there around neck and ear region. Okay? So all these are the symptoms of measles. Fourth one is another hepatitis virus, viral hepatitis. So there are different hepatitis. Okay? Hepatitis B, C. So this is caused by hepatitis B virus, viral hepatitis. The site of infection is our liver. Liver organ is infected, and mode of transmission is parental route. Genetically, it can not parental from blood transfusion, infected blood transfusion that is spread. Then, if a mother having this virus, then the born baby will also be that virus will be transmitted during the pregnancy. That is the parental route. The symptoms, liver damage, jaundice, nausea, yellowing eyes, okay, you will have fever, pain in the abdomen. So these are the symptoms. Viral hepatitis is that. Then another disease is chickenpox. Okay, varicella is the positive agent, zoster virus. So what is the site of infection? Is the respiratory tract. Respiratory tract is infected, and mainly skin and the nervous system is infected. The causative droplet infection, direct contact. Come to contact with a person with chickenpox, you can easily catch that. Then, what are the symptoms? Are mild fever with itchy skin, rash, uh, blisters. So, mainly skin is mainly damaged. A lot of itching sensation will go. It will be very uneasy condition for the chicken. That is the patient infected by chickenpox. Same thing, poliomyelitis. Now, this is poliovirus, virus group it is, where the intestine, brain, and the spinal cord is infected. So the site of infection of poliomyelitis, uh, positive infection, transmission is droplet infection through fecal oral route. Yes, so the mode of transmission. The symptoms are fever, muscular stiffness, muscular pain is there, weakness, paralysis, yes, the respiratory failure, paralysis. So you'll be having disorder. Muscle fatigue is there. Then dengue fever, breakdown viruses. So dengue, few years back, persons they were attacked by this dengue fever mosquitoes. So this dengue fever, dengue virus is a positive agent, flavivirus types. The site of infection, skin and blood is infected, and they said of transmission is through mosquitoes. Mosquitoes act as a vector. This chipti, this type of mosquito spreads this dengue fever. What are the symptoms? Are you will be having severe flu-like illness. Severe flu is there, then having fever, high fever, sudden set of fever, and painful headache, where you have severe headache, muscle pains, and joint pains. unbearable joint pains and muscle. Same is the case with the chicken, where it is called the alpha virus. It comes under the protovirus category. The nervous system is the site of infection. For this also it is mosquitoes only. That is HFT type. It acts as the vector. The, the symptoms are fever, severe fever, joint pain, unbearable pain, body pains will have severe headache, joint swelling, and so on. It has swelling in hands and legs. So these are the symptoms of chikungunya. Lastly, the COVID-19. Yes. So as you know, COVID-19 CO is for corona, VI is for virus, D is for disease. It was spread in 2000. That is the full form of COVID-19. If you look at the symptoms, you'll be having fever, loss of appetite, fatigue, okay, loss of smell, taste, and shortness of breath. If you're having problem breathing, continuous coughing is there, coughing of sputum. Sputum will be formed, muscle aches, muscle pains, you'll have severe muscle pains also. So this is common symptoms. If you see the severe symptoms of this disease, then difficulty in walking, confusion where brain is damaged. Yes. So bluish face or lips, then coughing of blood. When cough blood is seen, then persistent chest pain. They are having chest pain, decreased white blood cells, WBC will be decreased, kidney failure, and high. Yes. So these are the symptoms of this coronavirus. The main safety, primary basic safety is wearing a mask. That can be safe. Yes, with this I stop here. This class. So all of you who are present, switch on your videos so that I can take this. Exam point of view, you can definitely expect short answer structure of page. Then essay question, expect the viral replication process of virus. Both the cycles you have to explain, that is the lytic cycle and the isogenic cycle. As well as you may also have short answer like COVID coronavirus. Yes, there may